Trained as an immunologist at the Pasteur Institute, Paris, France, Dr. Steve Pascolo used mouse models to test and develop mRNA-based vaccines during his postdoc in Germany from 1998 till 2000. In 2000, he co-founded CureVac with Dr. Her and Dr. von Melby. Dr. Pascolo was chief scientific officer of the company from 2000 to 2006, developing the technology, implementing the worldwide first GMP production of mRNA, and performing the worldwide first clinical studies where humans, including himself, got injections of in vitro transcribed mRNA. In 2006, he joined the oncology department of the University Hospital of Zurich, Switzerland, and continued the development of immunotherapies based of RNA. In 2008, he founded Meteor Pharma to support this work, and in 2017, Dr. Pascolo implemented in Zurich an academic mRNA platform. In collaboration with several research and clinical departments in Zurich, he optimizes, tests, and implements mRNA-based therapies. Thank you so much, Professor Pascolo, for accepting to deliver this webinar. For all the participants, please feel free to write all your questions during the presentation in the chat box. You can use the language you want. You can write your questions in French, in Arabic, or in English, not in Chinese. We don't understand Chinese. So feel free to ask your question in the chat box. And it will be a very interactive webinar. We will address you some questions. We will interrupt the presentation from time to time to ask you questions. So, Mr. Dr. Pascolo, the floor is yours. You are muted, Steve.
Thank you so much. Thank you for the nice introduction. And thanks a lot for giving me the opportunity to present mRNA vaccines, and most particularly mRNA vaccines against cancer. Thank you to Foundation Diane for making it possible, this type of education, of seminar, of interactive seminar. And thank you also for the University Saint Joseph of Bayreuth. I will start, and as it was said, so you can interrupt me, we will speak about mRNA vaccines, how it works, the history of mRNA vaccine, a little bit about COVID, and then mostly about mRNA vaccines in development against cancer. My background is indeed academic and companies. Importantly, I started in 1998 in Tübingen to develop mRNA vaccines, and then I did it as academic and also as industry, being in CureVac, leaving CureVac. I have no more shares of CureVac since 2006. I'm not linked to CureVac anymore, but I have another company called Meteor Pharma, which is helping me to work with other companies. And mostly I'm academic in the University Hospital of Zurich and University of Zurich. So let's start with the science. mRNA vaccines have been the ugly duckling of vaccine development in the last 20 years. A lot of money and a lot of science was done on DNA vaccines, protein vaccines, viruses-based vaccines. We're getting the all attention and financial support, while the mRNA, by direct injection, the mRNA vaccines that we developed since the end of the 90s were disregarded. There was contempt, as you see from this deck, against mRNA vaccine. So we have been in the shadow of other types of vaccine development for 20 years. But suddenly there have been a pandemic in the per definition in the whole world, and a virus, SARS-CoV-2, coming from maybe from animals like this bat being eaten, or maybe from a leak from a high-level security laboratory where maybe coronaviruses escaped. We don't know what's the origin of the SARS-CoV-2 or the COVID pandemics, but this has changed totally the way mRNA vaccines are being recognized. Indeed, within less than 1 year from the publication of a sequence of a SARS-CoV-2 in January 2020, within less than 1 year, companies have developed mRNA-based vaccines against COVID, Moderna and BioNTech. The technology is BioNTech, but the clinical development was done together with Pfizer for the COVID vaccine. The vaccine resembles what is depicted here. It's synthetic mRNA here in green in a liposome, which is a bubble of fat consisting of 4 lipids, one of them being pegylated. So this is the representation of the nanoparticles contained in Moderna or BioNTech's vaccines. As an example of the efficacy of such mRNA vaccine, here are some data from BioNTech. So these are the neutralizing antibodies against SARS-CoV-2 in the serum of 10 people who got placebo injection but don't have any antibodies against SARS-CoV-2. On the other side, you have here the serum from people who have recovered from COVID, who have been infected by SARS-CoV-2 and who have recovered from the infection. They have neutralizing antibodies in their serum against SARS-CoV-2. And in the middle, you see younger people or elderly being injected with 10, 20, or 30 micrograms of the synthetic mRNA vaccine from BioNTech. And you see that with 20 micrograms, 30 micrograms, there are high antibody titers, neutralizing antibodies against SARS-CoV-2, even higher than the antibody titers obtained in people who have been infected and who have recovered. So this shows how strong is this mRNA vaccine at doses of 30 micrograms. This is very, very tiny. This is a very small dose of mRNA that is injected twice in people. If we go back in the past, there have been a lot of vaccines approved, and I divide them in 3 groups. There are the attenuated viruses, which are live viruses that are given mostly subcutaneous. And these vaccines infect you, and they deliver their nucleic acids in your cells, and your cells produce the protein of the vaccine and give an immune response. You have also dead viruses. So these viruses are usually given intramuscular. They are dead, they don't infect. They are usually given with adjuvant. And in third category is more advanced vaccines from molecular biology or from biochemistry. They are subunit vaccines. They are also given intramuscular, like the dead viruses, also with adjuvant. There are several platforms to develop new vaccines. You can develop them on DNA, RNA, or protein. In a cell, as a reminder, the DNA is in the chromosome. The DNA is stable. It remains in the nucleus of all your cells. It replicates. Parts of the DNA are transcribed into messenger RNA. The messenger RNA is transient. It stays a few minutes or a few days, depending on the mRNA in the cells, then it's degraded. The mRNA is translated into a protein, and the protein is also transient. It can stay a few seconds for proto-oncogene to several weeks, like antibodies, but at the end, it's also degraded. First question to you. What is the active ingredient in the vaccines against yellow fever, mumps, measles, or measles? Is it DNA? Is it messenger RNA? Is it protein?
Okay, everyone, so now you can see the poll in front of you, I guess. You will have some time to answer the question, and later on we will launch the answers so everyone can see what your opinions are. So
should I
again,
say the answer now, or we wait the end of the meeting?
No, doctor, people are still answering, so let's wait a little bit. We have a lot of people answering, it's good.
So we have 34 answers over 75. Come on. I can see.
Of course, we encourage everyone to answer and to be interactive.
Someone is writing on the PowerPoint, so please remove it. Yes, maybe we can share the results. We have enough answers.
Go to, I will now share the results. And here. We can see the percentage in front of us, so mainly the bulk of the participants think that the active ingredient is protein. Maybe the PowerPoint gave a hint. Anyway, 30% think it's mRNA and 17% is DNA. So doctor, why not enlightening us in the actual answer?
The right answer is messenger RNA. And I will show that on the next slide. But maybe the poll needs to get away from the screen.
The poll now.
You have to stop sharing, yes.
All good.
It's still on my screen. I cannot go to the
Yes, you should stop sharing the results.
I removed it. Is it still there?
It's still on the screen. And I cannot go to the
You
next
can
slide.
You can click on the X ups on the.
Okay. But my slide is frozen. Ah, now it goes. So the explanation is here. You see here the yellow fever virus, the mumps virus, the measles virus, the rubella viruses. They are attenuated viruses in the vaccines. They contain proteins, but they are RNA viruses. So their genome is not DNA, it's messenger RNA for all these four. And these vaccines are given sub-Q without adjuvant. In these conditions, the proteins in the virus, they cannot give an immune response. If you want proteins to give an immune response, you need an adjuvant and you need to get them intramuscular. Here, these vaccines, they are given sub-Q without adjuvant. So their active ingredient is the messenger RNA, the natural messenger RNA that they have, which goes in your cells when you get vaccinated. And this messenger RNA then makes the protein in your cells, makes the protein of a virus, and your immune system recognizes that. And this is important to know because everybody thought in 2020 that the mRNA vaccines are new. No, what is new is the synthetic mRNA vaccines. Yellow fever vaccine has nearly 100 years, and it is a natural mRNA vaccine. So when you get this vaccine, you get viruses with mRNA inside, and these viruses give you the mRNA in your cells, and your cells make the protein of the vaccine, make the protein and trigger the immune response. It's the same mode of action than you have with a synthetic mRNA vaccine. So it's a transfer of messenger RNA from here a virus, here a liposome, to your cells, and your cells make the proteins and make then the immune response. So all these are mRNA vaccines, but in the past we had natural mRNA vaccines, and now we have synthetic mRNA vaccines, which is purer, it doesn't contain proteins, it's only one mRNA and some liposome. I still have on my screen the red.
Yes, I'm sorry, I will ask Muhammad, I think he did it, if you can remove it, Celine tried to remove it. Otherwise, let's try to stop sharing and share again, because it's a colleague, Muhammad, he did it and he is not able to remove it. So if you can stop sharing, Steve, okay, the slide, and Celine, try to do something. Okay. So try to share again, please. Yes.
Great.
Yes. So please don't try it, for talking to the participants, don't try it on the slides. Thank you.
So yes, the right answer was mRNA is the pharmaceutical ingredient in these vaccines. It's natural mRNA. Now we have synthetic mRNA in our vaccines. And then other vaccines, all vaccines already contain DNA. So attenuated DNA viruses, like the vaccine against smallpox or chickenpox, are DNA viruses attenuated live. So they transfer their DNA in your cells when you get vaccinated. as we said, the mRNA, natural mRNA vaccines, yellow fever, mumps, measles, rubella, and many protein vaccines, for example, influenza vaccine, because here it is an old virus, but it's dead, it's killed by formaldehyde. So what is injected is with adjuvant intramuscular, and then it's the protein in the virus that trigger an immune response. So based on DNA, RNA, protein, you already have existing a few vaccines. And now in the context of COVID, all these formats have been used with modern technologies. DNA is the active pharmaceutical ingredient in recombinant adenoviruses. AstraZeneca, Sputnik V, Janssen from Johnson & Johnson are adenoviruses, recombinant adenoviruses. So what is injected is DNA in a virus, recombinant DNA. So these are DNA vaccines. The mRNA vaccines against COVID are from BioNTech and Moderna. And there are a few protein-based vaccines being developed against COVID, for example, from Novavax. They use protein, recombinant protein from the virus. Now let's focus on messenger RNA. Let me remind you again what is messenger RNA. Here is a cell, here is the nucleus of the cell. In the nucleus, you have the chromosomes. In the chromosomes, you have DNA. So the genes in the DNA are transcribed into pre-messenger mRNA. This pre-messenger mRNA is modified. At the 5' end here, it is capped with this molecular structure. At the 3' end, it's polyadenylated. It goes outside of the nucleus, and then it's further modified, and then it's being translated into a protein. In vitro, we can produce this molecule, this mature messenger RNA, and we produce it in two different ways, either non-modified or modified. Non-modified, it contains the 4 canonical bases of nucleic acids, of ribonucleic acid, ACGU. And this non-modified RNA, when you inject it, it's being recognized by some receptors of immune cells, mostly called Toll-like receptors. And these Toll-like receptors will immediately give a signal of inflammation. So when you inject synthetic non-modified mRNA, you immediately get an inflammation, which is an adjuvant effect in vaccination. So non-modified mRNA are used for vaccination against cancer, viruses, and allergies, for example, and in development. And when you modify the mRNA, when you produce pseudouridine mRNA instead of uridine, then this mRNA is not recognized by the immune receptors, the Toll-like receptors. So you can inject this mRNA without triggering an inflammation. So you can do gene therapy with this modified mRNA, expression of therapeutic proteins like erythropoietin, EPO, antibodies. You can reprogram cells. You can change genomes when you code for proteins like Cas9. Using modified mRNA. In 2017, however, it was found that you can also use modified mRNA surprisingly for vaccination, because since publication of this modified RNA in 2005 by the University of Pennsylvania, it was said that this is not immunostimulating. But finally, in 2017, University of Pennsylvania, Drew Weissman has published that still, surprisingly, with some formulations, this modified mRNA can also be used for vaccination. So nowadays, both modified and non-modified mRNA are being used for vaccination, as we will see in the rest of my talk. The production of mRNA is important to know that it's very fast, very easy to do. And that's why mRNA has been so quick to produce in the COVID pandemic. And that's why mRNA is being used against cancer, is because the production of mRNA is very easy. You just need a DNA, here depicted here, and the part in red is the gene of interest. It can be the spike of SARS-CoV-2, it can be a hemagglutinin from influenza, it can be a cancer protein, as we will see later. So you have this DNA starting material, and then you put this DNA together with the RNA polymerase that recognizes here a sequence before the DNA. You put the nucleotides, the cap analog here, and in a few hours, the RNA polymerase will make thousands of copies from this DNA. So you get also an amplification. So in the tube, you will get from one molecule of DNA 1,000 or even more molecules of messenger RNA within a few hours. And again, it's robust, which means it does not depend here on the sequence. Whatever is the sequence here in red, it will always work. It's like doing a photocopy. Here, the DNA is the book, and then you press 1,000 and you get 1,000 photocopy it's independently of what is on the book, whether it's a receipt for cooking or whether it's a poem, it's egal. You will always get your photocopies. That's what is happening when you do the so-called in vitro transcription. So you get your mRNA and there is never any problem. At the end of those 2 hours, you put DNase, an enzyme that destroys DNA, and then you can purify your mRNA, for example, by precipitation. So it's a very easy process in 4 steps, and it always works. The word robust is important. It was important to make the vaccine so quickly against COVID, and it's important to make vaccines against cancer, as we will see during the second part of my talk. Question to you. Is synthetic messenger RNA stable at room temperature?
Okay, so another question I will ask everyone to answer. And actually, it was a question that was asked in the comments, Doctor. How stable are synthetic mRNAs? What is the possibility for them to be degraded or maybe leading to making a different protein? So I guess we will answer this now. This was a question by Kausar.
Excellent. So you are more reactive now. We have like 55 answers.
Yes, 70.
So keep on answering. The more you answer, the more we have access to your presentations.
In the meantime, I have a question. Should I ask it now, Doctor? I have a question in the messages. It's in French. It's by Elia, whatever it is. The question is, un virus vivant atténué ne risque pas la transmission et la présence de nouvelles souches? Alors là, moi, je n'ai pas trop compris la question. Peut-être, docteur, si vous avez compris.
Yes, it's called reversion. There's a risk when you use a live attenuated virus that it will revert to the pathogenic virus. So that's the risk of live attenuated viruses is the reversion. So the live attenuated viruses that are used against mumps, measles, yellow fever, they have been developed since many years. We know they don't revert, but it's always a challenge. For example, in the SARS-CoV-2, people try to make attenuated viruses, but it's high risk because if your virus is still living and attenuated, you cannot be sure if you vaccinate millions of people, but suddenly it will not become again pathogenic. So that's a risk with live attenuated viruses, is that they would revert to become again pathogenic. That's why dead viruses are safer, but they are less efficacious. You need adjuvant. Live attenuated, you can inject them without adjuvant, sub-Q. So they are better. The yellow fever virus, the yellow fever vaccine, with one injection, you have a lifelong immunity against yellow fever. So that's very potent. But you're right, to play with live viruses in vaccines is having some theoretical safety concerns. And for SARS-CoV-2, nobody came up, as far as I know, with a live attenuated SARS-CoV-2 to be risky.
Thank you, Doctor. I hope we answered the question. And the results of your question are now on the screen. So mainly people think that no, synthetic mRNA are not stable according to our participants. So what's the answer?
So the answer is yes, it is stable at room temperature. And actually, mRNA, synthetic mRNA, is the most stable of the biological molecule. It's the only one that actually you can even boil or heat to 95 degrees. Of course, in the absence of RNAs, people think RNA is not stable because we have RNases everywhere, and the RNases degrade the RNA. The DNAs degrade the DNA. The proteinase degrade the protein. But without RNases, mRNA is very stable, and you can heat it to 95 degrees, and cool it down and freeze it and lyophilize it, it will never lose its structure and its functionality. On the contrary, if you heat DNA to 95 degrees, it will denature, it will lose its functionality. If you heat proteins to 95 degrees, they will denature, they will lose their functionality. mRNA is the only of its biological molecule that you can heat to 95 degrees, cool down, freeze. So yes, mRNA is actually very stable at room temperature. What is not stable so far in the vaccines is the formulation. The liposome that I showed you in yellow on the previous slide that encapsulates the mRNA, these are bubbles of fat, and these so far are not so stable. At room temperature, they may aggregate, they may change size or form and lose their functionality. They may release the mRNA and then the mRNA is lost. So that's why the vaccines against COVID from Moderna and BioNTech have to be stored frozen. It's not because of the mRNA. The mRNA is stable. It's RNA-free mRNA. The vaccine is very pure mRNA. But the liposomes so far that have been used in the vaccines are not so stable for such a long time at room temperature. Another question, is synthetic RNA in a synthetic liposome a pharmaceutical product that was never approved before 2020?
So here, maybe, doctor, we should add a little bit context, because some of the people were really scared of the vaccine, of the mRNA vaccines when it came up, especially arguing that it's a new technique and we do not want to be lab rats, basically. So this is what the question is about, I guess, that if it's true that it's actually a new technique, or was it used previously?
That's here the question. But there is a nuance in the question. I will, in the answer, I will tell you it's not an easy question. But yes, of course, in a way, the synthetic mRNA technology appears new for people, even if, of course, we work on it since more than 20 years and have done the clinical studies 20 years ago, started 20 years ago, mRNA vaccines against cancer. But people didn't know, for example, that mumps, yellow fever are also mRNA vaccines, natural mRNA vaccines. So that's why it's important to explain to people what are the components of previously licensed vaccines and very old vaccines like yellow fever, because when people know that it's mRNA, then they understand, okay, we change from a natural mRNA vaccine to a synthetic mRNA vaccine, but it's actually even purer and better. It's not so new, it's just the manufacturing is new, but the concept of vaccinating with mRNA comes already from yellow fever virus, attenuated virus, which is 100 years old.
Yes,
so here we have
somewhat, and I will share now the results so people can see the answers. It's kind of 50-50 actually, the opinions are...
still a small majority of no. So people think that RNA in liposome was never approved before 2020. They are wrong. It was approved, but not messenger RNA. But siRNA. Onpattro, also called patisiran, was the first drug based on RNA approved. It was approved in 2018. Again, it's not a messenger RNA, it's a siRNA. siRNA means small inhibitory RNA. It's synthetic RNA, but it's short, and it's actually allowing to destroy messenger RNA in the cell. So it's like the opposite of messenger RNA, but both are RNA. Messenger RNA is RNA, siRNA is RNA. Onpattro is a formulation of siRNA, of synthetic RNA, in a liposome, and the liposome of Onpattro is very close to the liposomes in Moderna and BioNTech vaccines. It's very similar liposome, and this drug, approved in 2018 on the market, is for patients suffering from a hereditary disease, the hereditary transthyretin-mediated amyloidosis. And in this disease, there is too much production of a protein called transthyretin. So this patient gets this synthetic RNA in the liposome intravenous at 30 milligrams every 3 weeks intravenous for their whole life. Remember that the vaccine from BioNTech is 30 micrograms. So here it's 1,000 times higher what these patients get their whole life every 3 weeks intravenous. Again, it's not messenger RNA. That's why my question had to be nuanced. My question was whether RNA in liposome was new. And the answer is no. mRNA in liposome is newly approved as vaccine. But RNA, in general, synthetic RNA, like siRNA, in liposome was already approved in 2018 and is given at 1,000 times higher dose intravenous to these patients compared to the vaccines. Last question on this topic, is synthetic mRNA vegan? Or does it have animal products? Does it contain animal products or is it free of animal products? I think that's a clear yes, and you're right. The mRNA vaccines, the synthetic mRNA is vegan. The production I showed you on the previous slide use enzymes like RNA polymerase, which are produced in bacteria, and nucleotides, which are chemically produced. So the whole in vitro transcription and purification does not involve animal products. So it's a good thing for vaccine to be vegan because it's better for the environment, which is also important for the GIAN Foundation. So you have a less impact on the environment by producing a synthetic mRNA vaccine compared to other kind of vaccines that need to be produced using animals. For example, eggs. The influenza vaccine every year is produced in eggs. So you need millions of chicken to make millions of eggs every year just to produce the influenza vaccine. So of course you have an impact on animals and on the environment producing the normal influenza vaccine. It's not the case with the synthetic mRNA vaccines. They are vegan, and also it's an advantage medically because people who have some allergies against food, for example, there is no problem with the synthetic mRNA vaccine. People who have allergies against eggs, they cannot get the flu vaccine because it's produced in eggs. So here with the synthetic mRNA, you have less problems with allergies. There are still existing some cases of allergies against the synthetic mRNA vaccines against COVID, but it's very rare because it doesn't have animal products. So finally, one last point on the production of synthetic mRNA. You need very small infrastructure to produce synthetic mRNA vaccines. In 6 liters of in vitro transcription, so the DNA, the RNA polymerase, and the nucleotides, if you have 6 liters of that, after a few hours, you will have enough mRNA in the 6 liters to make 1 million of doses of the vaccine. And when you want to compare to virus-based vaccines, for example, when you produce a measles virus or this kind of virus in cell culture, then you need like 1,000 times bigger infrastructure to produce 1 million of doses, like 5,000 liters, 6,000 liters of culture are needed to produce enough virus for 1 million of doses. So one advantage of the mRNA vaccine is that you can produce it in relatively small infrastructure. And this is also important, as we will speak about, for cancer vaccines. A few words on the history of mRNA vaccines. It's a real European history. It's actually a French-German history. mRNA was the natural molecule mRNA was discovered in 1961 in the Pasteur Institute in Paris by François Jacob and Jacques Monod, who got the Nobel Prize in 1965 for their discovery of the messenger RNA. And then still in Paris, in 1993 was the first publication, worldwide first publication of using synthetic mRNA as a vaccine in animals. It was the Professor Pierre Melien in Paris who had the idea to produce synthetic mRNA coding for influenza protein, to put it in a liposome. So the formulation that Professor Melien published in 1993 is very similar to the formulation we have 30 years later, synthetic mRNA coding for virus protein in a liposome. And Professor Melien's team could show that when they inject the synthetic mRNA in a liposome in mice, the mouse develops an immune response against viruses. So discovery of mRNA and utilization of synthetic mRNA as vaccine, both in Paris. And the first clinical studies we have done actually in Tübingen in Germany. We have produced synthetic messenger RNA coding for luciferase. Luciferase is a protein that allows the firefly to emit light. So we produce mRNA coding for luciferase, and I got injected several times with this mRNA to show that first the mRNA was safe in human, the synthetic mRNA injection were safe in human. I didn't have local or systemic side effects following these injections. And then we could detect in biopsies made in my skin at the site of mRNA injection light emission, meaning that the synthetic mRNA coding for luciferase from the fly could be expressed in human skin and without any side effects. So I did express actually these fly proteins and transiently and without side effect. Then we also did the first clinical studies, the worldwide first clinical studies with synthetic mRNA. It was against cancer, starting in 2003. We started in the dermatology clinic where also these experiments were done in the University Hospital of Tübingen. We injected synthetic mRNA vaccines to melanoma patients wanting to trigger immune response against melanoma, as I will show you a little bit later. And during the 20 years, or over 20 years of development of mRNA vaccine, only by a few people in the world, very little people were doing that. It was the ugly duckling of vaccines, but there have been 20 years of improvement, in big part performed by the great team of Ugo Sahin and Özlem Türeci in Mainz, also in Germany. Both things were done in Germany. And this wonderful family with the baby reading Goethe is an extraordinary family who has led the development and improvement of mRNA vaccines and allowed during the pandemic of COVID to get approved the first synthetic mRNA vaccine, actually the mRNA vaccines against COVID from BioNTech in collaboration with Pfizer. Now we move to cancer. So before we move to cancer, any questions about synthetic mRNA production, formulation, COVID?
Docteur, on a une question dans le chat de Tala. Elle demande quels sont les critères qui déterminent le mode d'injection d'un vaccin, que ce soit intramusculaire, sous-cutané ou intraveineux.
Docteur Pascolo, si c'est possible, quand la question est en français, de répondre en français pour être équitable avec tout le monde.
OK. C'est l'expérimentation qui permet de savoir, en général, sous-cutané est réservé aux virus vivants, atténués, et intramusculaire à tout ce qui est mort, virus mort, protéines, subunits. Donc ça, c'est une guideline générale. Maintenant, par exemple, dans le contexte des vaccins ARN messagers, ils sont utilisés intraveineux dans les cas de cancer, on va le voir tout à l'heure. Ils sont utilisés sous-cutanés pour l'instant avec relativement peu d'efficacité chez les animaux. Et les formulations certaines formulations sont intramusculaires, comme celle validée contre le Covid. Donc c'est une bonne question parce que, en général, donc dans le passé, c'était sous-cutané pour les virus vivants dans les vaccins, intramusculaire pour ce qui était mort. Donc il y avait à peu près ces guidelines qui étaient claires. Pour le mRNA, pour l'instant, cette formulation fonctionne bien intramusculaire, mais on travaille sur de nouvelles formulations d'ARN messager synthétique qui pourraient bien fonctionner aussi sous-cutanées, parce que les injections sous-cutanées donnent moins d'effets secondaires, donc on essaye de développer des formulations, ça dépend beaucoup du liposome, et on essaie de développer des formulations d'ARN messager qui fonctionneraient sous-cutanées. Pour l'instant, celles qui sont sur le marché de Moderna et de BioNTech fonctionnent en intramusculaire. Il faut continuer à les injecter en intramusculaire. Il faut développer des nouvelles formulations d'ARN messager synthétique qui vont, j'espère, être aussi efficaces en sous-cutané, ce qui permettra voilà, peut-être d'avoir moins de réactogénicité locale dans le muscle après l'injection. Mais c'est une bonne question. Il y avait les guidelines, comme je disais, live attenuated, des virus vivants sous-cutanés, tout ce qui était mort intramusculaire. Maintenant, l'ARN, pour l'instant, est intramusculaire, mais je pense qu'on va réussir à avoir des formulations dans le futur qui seront aussi bonnes en sous-cutané, ce qui est un petit avantage pour les gens d'avoir une sous-cutanée plutôt qu'une intramusculaire.
Thank you, Doctor. We have another question by Bilal. Why do some people develop symptoms of the disease after the vaccination and others don't?
Yeah, that's very complex. You know, it's the same question you can ask, why do some people get sick with SARS-CoV-2 and others not? It really depends on your genetics. It really depends on your immunological memory. in our life, we all met some coronaviruses, so we all have some antibodies and some T cells against coronaviruses, not the SARS-CoV-2, but other types of coronaviruses. So depending on your genetics, on your immune fitness, on your immune memory, you may respond stronger or less. And for the time being, there is, as far as I know, no prediction of what makes people having a strong reactogenicity and side effects to the vaccine, to the COVID vaccine versus those who do not have. Normally, younger people, it really depends. So, and it's not explainable. It's a combination probably of factors, as I said, your genetics, your fitness, your immune fitness, because when you get elderly, your fitness is, your immune system is lower. So the fitness of the immune system and the memory of the immune system, how much of memory immune response you have against coronavirus that may cross-react with vaccine. So all these parameters make some people feeling nothing after the vaccines and some people being quite sick, especially after the second injection, but it's only one day. So as far as I know, people don't know and cannot predict whether the COVID vaccine, the synthetic mRNA vaccines against COVID will give you strong or not strong side effects. It's being studied.
We have more questions. Actually, Doctor, would you like to continue or, and ask the questions later, or do you prefer answering all the questions before moving on?
As you prefer. I think maybe these questions are about COVID and vaccine. It's good to answer them, but if you prefer to go to cancer, we can go to cancer. You decide. Yes,
because we're receiving, we are still receiving questions, so I think it's better to move on and we will answer later, just for the sake of having enough time.
Okay. Okay, good. So we move to cancer. So you can vaccinate against cancer. You can train your immune system to recognize and kill cancer cells. And there are two ways to immunize against cancer. They are so-called share antigens. So these are antigens, mostly proteins, that are expressed in many cancer cells and many cancer cell types. There are several families of these so-called tumor-associated antigens, TAA. So they can be cancer testis or germline antigens. They can be overexpressed antigens. So these are proteins that are expressed in human. The cancer testis antigens, they are usually expressed during embryonic development, and then they are not expressed anymore in the normal body, except in the testes, but they are re-expressed in cancer. And cancer cells also overexpress some proteins, like HER2 is a protein that is highly expressed in some breast cancers, for example, and there is an antibody called Herceptin that recognizes HER2. So HER2 is a normal protein expressed in many cells of our body, but it's overexpressed in many cancers. And so you have these families of TAA, tumor-associated antigens. So you can prime or activate your immune system against this TAA in order to recognize tumor cells. There is another possibility. These are tumor-specific antigens.
Steve, you are muted.
Somehow the connection got lost and was reestablished.
Okay.
Do you see my screen, or should I do?
You should reshare your screen, please. Thank you.
Do you see my screen?
Yes.
Yes.
Okay. So these tumor-specific antigens, there are these three families: virus proteins, when the virus is responsible for the oncogenic feature of the cell, or these mutations. So, and there are mutations which are frequent in several cases of cancers, and there are mutations which are specific in each patient. And so you can vaccinate with this against these mutations of this protein. So here it's called tumor-specific antigens. I will say in the rest of my talk TSA, and this is TAA. So you can vaccinate against these two types of antigens and then get an immune response that can, in some ways, recognize cancer cells. Questions? Do all cancer cells contain mutations? Okay, so we are at 50-50 nearly. So yeah, it's 50-50 nearly. So, but with a small majority of yes, and yes is right, yes is correct. All cancer cells contain mutations, but it's not the same frequency of mutations depending on the type of cancer. Some cancers here, like melanoma, have a high frequency of, so we are really at 50-50, have a high frequency of mutations here in the number of mutations per megabase in the genome. So people sequence melanoma, and in average, you have in melanoma quite a lot of mutations that you can record in the melanoma cells compared to healthy tissues. But you see that in some cancers, like sarcomas, like glioma, you have much less mutations, and these cancers are most of the time harder to treat with vaccines or with immunotherapies, while this type of cancer, melanoma, lung cancer, bladder cancer, they respond well to immunotherapy, maybe because they have more mutations, which makes them more recognizable by the immune system. Because of the recognition of this mutation by the immune system, probably these cancers are more sensitive to immunotherapy, to vaccinations or immunotherapy, so interventions with the immune system. While some cancers, maybe because they have less rate of mutation, are harder to treat with immunotherapies. But all cells, even if it is low, all tumor cells have mutations which make them tumor cells. Based on these two formats, TAA or TSA, as I said, you can vaccinate. And we started to vaccinate with mRNA against both TAA and TSA, and actually combine them. And this was the program of Professor Ugur Sahin from BioNTech. It was started in 2012. We got EU support and made a consortium called MERIT. And this support was to actually do this kind of treatment in cancer patients. So you take a biopsy of the cancer of a patient and you sequence it in order to analyze the mutations. During this work, you immediately inject the patients with some mRNA coding for shared antigens that you have off the shelf. So you have a bibliothèque of mRNA coding for shared antigens from cancer, MAGE-A1, NY-ESO1, HER2, etc. You have a bibliothèque, and you can immediately inject the patient with this mRNA coding for TAs. And during this time, you sequence the cancer of the patient, you identify the mutations. It takes 3 months, and then you make mRNA coding for the mutation sur mesure. It's haute couture of messenger RNA vaccine. It's haute couture of vaccine. You make for each patient a vaccine that encodes the patient's mutation. So during a few months, the patient gets messenger RNA vaccine that codes for shared antigens, and then the patient gets mRNA vaccine coding for its mutation found in his own tumor. So this is called MERIT, and it was coordinated by Professor Goursahin. We are involved in Zurich. We were involved in this network, and this kind of approach continues. In the past, we had done already several publications on messenger RNA vaccine against cancer. The first messenger RNA vaccine tested in human was a messenger RNA vaccine against cancer. That's the clinical study that I published in 2008. And then there have been a few other mRNA vaccine being published in 2009, 11, and one by Professor Goursahin. We will go quickly through the data from our 2008. I want to show you how these have improved, how the mRNA vaccines against cancer that were started in 2003 have given some promising data, but the mRNA vaccines against cancer that we have now are much better. So let's start with the old results published in 2008 in melanoma patients. We injected the melanoma patients with mRNA coding for TEA and TSA, actually a mixture. And we could see here, for example, in 4 patients, when you look with time, their T cells against cancer, recognizable here as the dots in the upper quadrants. Over time, you see that the frequency of the anti-tumor T cells increase in patient male 02, in patient male 03. So we could show in some patients that their CD8 T cells or CD4 T cells get against cancer have been increased during the course of vaccination. It's not strong, we could still detect it. And here is the antibodies against cancer. The background is the line, and the antibodies against cancer are the black histogram. You can see over time in some patients that we can see antibodies developing against cancer. So it's not in all patients, it's not strong, but we could see that the vaccines, the mRNA vaccines in these cancer patients induce some immune response. Clinically, we could see most patients progress, unfortunately, but one patient had a stable disease during 6 months during the time of vaccination, and some patients with no tumor, because they have been operated, remain tumor-free during a few months under vaccination. So these were the clinical data, the very first, worldwide first mRNA vaccine trial, anti-melanoma mRNA vaccine. A third trial we have made, the second one was also in melanoma patients. The third one was in renal cell carcinoma patients, also in Tübingen. Here you see again in some patients that there's an immune response that was induced over the time. So before vaccination, you see barely any immune response against the proteins encoded by the mRNA. This was a mixture of 6 mRNA coding for cancer proteins of renal cell carcinoma. And you see over time that then the patients develop a T cell response against their cancer, against these antigens that are in the vaccine. So indicating that the vaccine has worked. It has induced T cells against the tumor proteins that are coded by the mRNA vaccine. Clinically, we could see some patients having partial response in their clinical development. Some patients getting stable. So most patients had a stable disease during 12 months. So their tumor was stable. They didn't progress. And one patient here had a partial response, which means that the tumor regressed during the time of vaccination. So we had some clinical response in terms of stable disease or partial response, but most patients progressed. So this was again published 2011, more than 10 years ago. Now, Professor Goursahin in Mainz has developed, as I said, in the context of MERIT, this individualized vaccine, haute couture, mRNA vaccines made for each patient. And this now he published in 2017. These are melanoma patients. These are 4 different patients over time. They are monitored. Again, here, the quadrant shows the T cells that are induced by the vaccine. You see at the beginning, you have little or no T cells that can recognize the proteins, or here the mutations actually coded by the mRNA vaccine. And along the time, the patients get injected with the synthetic mRNA vaccine coding for the mutation, and they develop T cells against the mutations that are that have been identified in their cancer. These are totally individualized mRNA vaccines. And again, these individualized mRNA vaccines are possible because the production of mRNA is so easy that you can really make for everyone his own mRNA vaccine. It's not so easy if you do peptide or proteins. With mRNA, as I showed you on the production slide, it's robust. So you can always produce mRNA very easily in small scale for every patient. And here, Professor Sahin has shown that these mRNA vaccines coding for mutations work. You have T-cell response that develop thanks to the vaccine. And clinically here, you can see in all these patients, in the last 24 months, they had several metastases appearing and being removed by surgery. And after vaccination, in most of these patients, there have been no more new metastases. So they have been tumor-free, no evidence of disease. Many of these patients, also before vaccination, they had quite a lot of relapse. During vaccination, they had less relapse, indicating that these T cells induced by the vaccine could protect the patients against relapse of their cancer disease. And a last publication I want to show, also from Professor Ugur Sahin and Özlem Turecci, this time using tumor-associated antigens, so not the haute couture mRNA vaccine coding for mutation, but mRNA coding for shared antigens. Here they have used either the mRNA vaccine alone or together with immunotherapy, the so-called immune checkpoint inhibitors, anti-PD-1 antibodies. And you see that in quite some patients here, their tumors regressed. So the limit is 25% regression. That makes a partial response. And you see quite a few patients with a vaccine alone as monotherapy here in dark green, or with a vaccine plus anti-PD-1 immunotherapy have regressed. So their tumors regress, shrunk to less than 25% using the combination of vaccine and anti-PD-1 or the vaccine, the synthetic mRNA vaccine alone. So this shows you how in 20 years, we have started from formulations a little bit less efficacious to now formulations more efficacious with more clinical responses. And I will now shortly update you with the current situation. Leads are still BioNTech and Moderna. BioNTech has several clinical studies ongoing. FixVac are mRNA vaccines coding for shared tumor antigens. iNeST are mRNA vaccines which are individualized, haute couture. Every patient gets its own mRNA vaccine. It takes 3 months, but then it gets its own mRNA vaccine coding for the mutation. So there are several clinical studies ongoing in different types of cancer, and including melanoma, but also all types of solid cancers. There are a few studied in phase 2, so we can hope that in the near future, this type of mRNA vaccine against cancer could be approved. So here, the mRNA vaccines against cancer from BioNTech, they are given intravenous. They are liposomal formulation given intravenous, and they are non-modified mRNA in all the cases. The Moderna vaccines against cancer, they are modified mRNA. They are given intramuscular, and one is in phase 2, one is in phase 1. They are both for the coding for the tumor-specific antigens, mutations, and this is, again, ongoing. I will update you on some of these trials. Here is just a table of some clinical studies using mRNA vaccines in melanoma, lung cancer, ovarian, prostate. I will not detail it, it's just to give you an impression. There are many clinical studies that have been done and that are ongoing. Lead is BioNTech and Moderna, but other companies also and universities are performing clinical studies. If we focus on the most advanced, it's this MERIT from Professor Ugur Sahin. It's in phase 2 in melanoma. And the recent communication about the status of this trial has shown that, so this is an injection of mRNA coding for tumor-associated antigens, so no mutation, just normal non-mutated proteins expressed in tumors. NY-ESO-1, MAGE-3, tyrosinase, and TPTE. So these are 4 mRNA coding for these 4 proteins injected intravenous. And in 89 patients, this protocol was used with a dose range from 7 microgram to 400 microgram given intravenous. Keep in mind that the COVID vaccine is 30 micrograms intramuscular. And here the recent data has shown that 3 patients out of 25 achieved partial response. So it's like more than 1 in 10 patients have achieved a partial response, and 7 have achieved stable disease, and 1 had a complete remission. So you see here quite high rate of clinical responses induced, or at least during the time of vaccination, much higher than what we had and we published in 2008, because the mRNA vaccines are better now, so they induce stronger immune response and stronger clinical responses. The same is true for the BioNTech vaccine, coding for tumor-specific antigens mutations. They've shown that at a 8-month follow-up, 11 of 13 patients remain disease-free. So these were disease-free patients who got operated, the tumor was removed, and they got vaccinated, and they didn't have a relapse, they didn't have a recurrence, these patients, 11 of these patients. And in patients who had tumors that were not removed because they could not be removed by surgery, this mRNA vaccine was combined with immune checkpoint inhibitors, PD-1, and 1 out of 20 patients had a complete remission, and 5 had partial responses. So 1/4 of the patients had partial responses. In our first studies, we have 1 in 20, 1 in 30 patients who had a partial response, and now it's 1/4 because these formulations are so efficacious. Just to finish, 2 last update on some clinical studies. This one from Moderna, again, monotherapy or combined vaccine, mRNA vaccine with PD-1 treatment, so immunotherapy, and up to 1 milligram given intramuscular, and coding for mutations in this case. And here in 10 patients of a subgroup, 2 patients have achieved complete response and 3 achieved partial response. So like 1/3 of the patient achieved some partial responses, and 1/5 complete responses. Here another BioNTech vaccine study, and here it's again intravenous non-modified mRNA coding for mutations, individualized, so haute couture mRNA vaccine. Every patient gets its own vaccine, and of 26 evaluable patients, 1 patient had confirmed complete remission for more than 10 months, and 12 patients achieved stable disease. So nearly half of the patients did not progress, they stayed stable during the vaccination. So all these are very promising, and I will finish with these conclusions. But vaccine against cancer, whether the mRNA tumor-associated antigens or whether the mRNA codes for mutations, are more and more efficacious. When you compare the results we had in 2005 and the results we have now in 2020, 21, the mRNA vaccines are much more efficacious. The immune response is much stronger that is induced by the vaccine. And then the clinical response rate is much higher, with some complete remissions or partial responses and some stable disease in many treated patients. And these mRNA vaccines against cancer, whether against TAA or TSA, are now combined with other therapies, most of the time immune checkpoint inhibitors, so anti-PD-1 or PD-L1 antibodies. So this is very promising. As you have seen, it's really moving to be better and better, and we can hope that in the near future they will be approved and they will continue to be improved. The mRNA, the formulation, and the targeted antigens, all this can be improved to have always better and better immune responses induced by the mRNA vaccine to control and prevent, so to control tumor growth and prevent relapses. We didn't speak about therapies with mRNA. It would be maybe the occasion of a next seminar, because with mRNA, synthetic mRNA, you can vaccinate, but you can also not vaccinate, just induce protein expression. And this is used with synthetic mRNA. For example, synthetic mRNA coding for anti-cancer antibodies or anti-cancer cytokines like IL-12. And these mRNA sometimes are given in the tumor so that the tumor produces its own anti-tumor protein. And this is fantastic, and this is also very promising. It's in clinical studies. And also cell therapies, where you take the blood cells of the cancer patient, you modify them with mRNA coding for chimeric antigen receptor, CAR. These are receptors that kill tumor cells. So in this way, you can use the mRNA to transform T cells into tumor killers during a few days. And this had been already published in 2014 by Carl June, and this was showing some efficacy. This is also being developed. I developed it also here in Zurich. So you can take blood cells, transfer in the blood cells synthetic mRNA coding for anti-tumor proteins, reinfuse the blood cells and get anti-tumor activity during 2 or 3 days. So this is therapy. It's not vaccination, it's therapy. And so we can speak maybe in the next seminar about synthetic mRNA-based anti-cancer therapies. But again, for synthetic mRNA-based anti-tumor vaccines, you have seen how it has started with low response rate, low immune responses induction, and now thanks to the progress in mRNA in formulation and in the knowledge about the tumor antigens in cancer cells, we can now have mRNA vaccines that are quite efficacious against cancer, and that will be more and more efficacious in the near future with more research, more clinical studies, approval of the first versions of these vaccines, and improvement of these vaccines. With that, I thank you for your attention. You can find more information about mRNA vaccines in the recent review I wrote and in reviews from others. We have support from Switzerland and from EU and from foundations for our work. And I welcome any questions. I'm happy to answer questions now.
Thank you so much, Professor Pascolo, for this amazing webinar. We gave us hope and a promise to continue this series of webinars with you. So I will ask you please to stop sharing the slides. Maybe we can open our cam, if you agree, dear participants, so we can discuss all together with Professor Pascolo for a few minutes. I will ask Celine to start asking you the written question in the chat box, and then we'll open the discussion. But it would be wonderful if you accept to open your cam so we can discuss all together. Thank you. Celine?
Yes, so I will go in order, basically. On a une question en français. Comment pouvez-vous surveiller le ARNM synthétique après injection dans le corps? Et quand il y a une réaction adverse, qu'est-ce qui s'est passé à cause de ce vaccin synthétique ARN?
Pour la première question, oui, ça a été fait. On a suivi, des gens ont suivi l'ARN synthétique dans les vaccins anti-Covid après injection. Donc, vous les trouvez, par exemple, dans le sang. Donc, il y a des liposomes qui vont ne pas rester dans le muscle, qui vont aller dans le sang. Donc, si vous prenez du sang des patients, des personnes, pardon, comme nous, injectées par le vaccin anti-Covid, vous allez pouvoir trouver pendant quelques jours dans le sang des traces de l'ARN messager synthétique. Donc, par PCR, par des méthodes très, très sensibles, vous pouvez trouver pendant quelques jours, je crois, jusqu'à 3 jours de mémoire. Donc, il est possible de détecter dans le sang de l'ARN messager synthétique, celui qui a été injecté pour vous vacciner, pendant quelques jours, parce qu'à la fin, l'ARN messager va être dégradé. Celui qui est local dans le muscle va être dégradé. Celui qui a été dans les nœuds lymphatiques, où il a induit la réponse immunitaire, va être dégradé. Et celui qui se retrouve un peu partout, en très petite quantité, va aussi être dégradé. Les gens ne l'ont pas trouvé dans le lait, lorsque les femmes qui allaitantes ont été injectées par les vaccins anti-Covid, Moderna ou BioNTech. Dans le lait, il n'y avait pas de trace de l'ARN messager synthétique, mais on le trouve dans le sang seulement quelques jours, puis on ne le trouve plus. L'ARN messager est toujours dégradé à la fin. Une protéine, c'est pareil. L'ADN, c'est différent. L'ADN des adénovirus, AstraZeneca, Sputnik V, Johnson & Johnson, théoriquement, pourrait rester longtemps, parce que c'est de l'ADN. L'ARN messager, lui, même s'il se retrouve un petit peu en périphérie, en toute petite dose, parce que ça a échappé du site d'injection, ça va de toute façon, après 2 ou 3 jours, être complètement disparu et ne plus être détectable. Donc voilà, on détecte éventuellement l'ARN messager synthétique un petit peu dans le sang, quelques jours, et puis il a disparu. Et la deuxième question, c'était les effets secondaires, à quoi ils sont dus. C'est beaucoup étudié. Donc, c'est dû. Alors, ils sont surtout présents à la deuxième injection. Et donc, ils sont dus en grande partie à la mémoire immunologique. La première injection induit très fortement une réponse immunitaire contre la spike de SARS-CoV-2, qui est codée par l'ARN messager. Et lorsqu'on fait la deuxième injection, il y a les cellules mémoire qui réagissent, qui produisent ce qu'on appelle de l'interféron gamma. Et donc, vous pouvez détecter dans le sérum des personnes après injection, après le boost, vous détectez de l'interféron gamma pendant 1 ou 2 jours. Et cet interféron gamma va faire une stimulation immunitaire systémique qui va donner de la fièvre, de la fatigue. Et donc, selon votre réponse interféron gamma, vous allez avoir de la fièvre, de la fatigue pendant 1 ou 2 jours. C'est la réponse immunitaire. C'est étudié, les mécanismes sont vraiment étudiés maintenant en plus de détails. On voit les cellules T et les cellules NK qui, après l'injection, après le boost, font de l'interféron gamma. On trouve cet interféron gamma dans le sang. Donc maintenant, les gens commencent à comprendre un peu mieux ce qui se passe, pourquoi vous avez des effets secondaires. Mais par contre, ça ne permet pas de prédire quelle personne aura des effets secondaires et laquelle n'en aura pas.
Ok, deuxième question en anglais cette fois. What about the interaction between spikes A and the ACE2 inflammation downregulator enzyme? Is it a toxic interaction?
That's a little bit out of my scope, so I'm not sure I would be the best person to answer. There might be some activities of a spike protein on certain cell types and certain cells. Now, we will all get infected, or we will all get in contact with SARS-CoV-2. And when you get infected with SARS-CoV-2, it will happen to all of us, or it happened already, then you have a spike protein everywhere in your body, because the virus will be everywhere in your lungs, it will be in your blood, it will be even in your reproduction organs. Everywhere in your body, you will have the spike, because you will have a virus. So if the spike can be toxic, then the problem is if you are infected, because then you have the spike everywhere in your body. When you are vaccinated, you have the spike locally in the muscle, in the draining lymph node, and maybe a little bit somewhere else, because some liposomes will go away from the injection site, but you will have the spike very transiently and mostly at one site. When you get infected, you have a lot of spike everywhere in your cells from your lungs, your cell from the blood. They will make virus, they will make spike, so you will have spike everywhere. So if spike can be toxic, then you should be afraid of a virus, because this will make you full of spike in your whole body, while the vaccine will give you spike locally and transiently.
Okay, third question. What is the strong level of antibodies for COVID and the weak ones? So what are the strong and weak levels of antibodies for COVID?
That is not possible to answer, because there are like 300 diagnostic tests to measure antibodies against SARS-CoV-2. And each of these tests has its own scale and its own readout. So this ELISA test, there is no harmonization. So there is not one value. Each test will tell you. So when the labs do the test for you, if they measure your antibodies in your serum, they will give you a report with a value, and they will tell you if you are positive or negative, or probably positive or probably negative. But if you do the test with 2 different kits, you may have different response. So unfortunately, people now spend some time and some efforts to take their blood to measure their antibodies. In the normal population, it doesn't give you an indication. It's useful for immunosuppressed people, because immunosuppressed people, they don't respond well to the vaccine. So you can measure their antibodies with one test along the time in order to see after how many vaccinations they start to have antibodies. Because for some immunosuppressed people, you need 3-4 vaccine injections before you get antibodies. So this is important in these immunosuppressed people, cancer patients, immunosuppressed people, to measure their serum antibodies with one kit in one lab. But there is no value worldwide that one can give to say you have antibodies. It really depends on which test you use. And even if you use one test, you may have varying values. So that's the answer.
Okay, yeah, next question. Why vaccines need boosters when we have memory T cells?
Because the T cells and the B cells, they mature, so they get better and better with time. So they make antibodies which have a higher affinity, and especially when they meet again the antigens, it's called affinity maturation of the T cell receptors and antibodies. So against COVID, it was quite clear that after the first injection, you have antibodies. that you can measure in the ELISA, but they are most of the time not neutralizing. And only after the boost, everyone has neutralizing antibodies. That's why, to bridge it to the previous question, when you measure in ELISA the antibodies, it also doesn't tell you if they are neutralizing. So that's really an information that is not useful for the general population. Again, it's useful only for precise patients when you want to follow them and to make sure that they have responded to the vaccine. Because what is important is to have neutralizing antibodies, and this appear really only after the boost, probably because the boost allows the best antibodies and the best T cells to get reactivated and to further get better along the time. It was shown that even in the months following the second vaccination, the memory B cells have better antibodies than the B cells that you had before. So they continue to select the best antibody against the virus.
Okay, someone wants to know what's new about vaccination of children under 12-year-old.
Yeah, for COVID now it has been approved. The mRNA vaccines against COVID have been approved for children. BioNTech is using 3 times less the dose. So adults, we get 30 microgram of BioNTech vaccines, and I think it was approved for children 10 microgram, 1/3 of a dose. Moderna, I don't recall for children if they have also lowered the dose or not. So now both vaccines are approved in the US for children, and I think also in Europe. So this is approved. It's not an easy decision, I think, for parents to decide whether their child should be vaccinated or not. In many countries, if you are not vaccinated, then you have restrictions to where you can go, where you can travel. So people vaccinate their children. It's a difficult question. So because children, most of the time, they don't suffer from COVID. They get infected and don't suffer from it. So they don't get the disease. So why should they be injected if they don't themselves get COVID? On the other hand, you have also some children who have been sick from COVID and who have died from COVID. So they are rare, but it exists. So you want to avoid that by vaccination. That's a good thing. You want to avoid that the children get infected or that they get that they can spread the virus. So for that reason, it makes sense to vaccinate children. On the other hand, it's a bit strange also to vaccinate children when they have no high risk. So it's really a hard decision. So now the vaccine is approved. The mRNA vaccines, both Moderna and BioNTech, are approved for children of less than 12 years old. Again, with a reduced dose, at least for the BioNTech vaccine. And then it's about the parents to decide. Again, if a child gets infected with SARS-CoV-2, he will produce a lot of virus, he will produce a lot of spike. He may not notice it, but he will produce virus, he will produce spike. So it's better to have him vaccinated so that it's eventually protected against infection or can solve the infection quicker than if it was not vaccinated. And especially if children have an environment of fragile people, then it's important that the kid doesn't bring the SARS-CoV-2 at home or has less risk to bring it at home. It's really a question. It's a difficult question for the parents. Of course, it was clear to vaccinate elderly, to vaccinate fragile people, to vaccinate adult people like me. I got vaccinated immediately when I could because I know what is in the vaccine. I know it's safe and it's good. So, but for children, it's still, I understand the balance of risk to benefit is a little bit harder to evaluate. I think the kid can also, maybe 11 years old kid can say if he's afraid of a vaccine or not, and if he's not, then he should get vaccinated. If kids have phobia of injection, that's a difficult question. So whether they should be vaccinated or not. But at least the mRNA vaccine are approved for children of less than 12 years old in the US and in Europe.
Okay, thank you. Now we have 2 questions that are a bit related. First, someone is asking, is the technology you described used on all kinds of cancer? And someone else is asking more details about the relationship between this vaccine and the breast cancer. So I guess you could answer them both.
Yeah, the mRNA vaccines against cancer, they are used against a lot, or they are tested against a lot of different type of cancers. Melanoma is most advanced, but there are also a lot of solid cancers. So there are clinical studies that recruit people with solid cancer. So it can be nearly any type of solid cancer, excluding lymphomas and leukemias. So solid cancers can be breast, colon, skin, anything. So a few mRNA vaccine studies, when they are targeting mutations, they can include any patients, because as I asked in one of my questions, do any cancer cells have mutations? The response is yes. So when you make mRNA vaccine sur mesure, then you can target any cancer because you have to define the mutation in any person, whether it's a colon carcinoma, a pancreatic carcinoma, lung cancer, then you can always identify the mutation, make an mRNA vaccine coding for the mutation, and then inject to this patient. So that's why a few of these mRNA vaccine studies recruit very broad, solid cancer, and a few are more dedicated to melanoma or breast cancer, especially if the mRNA codes for shared antigens. It's better to be in one type of family of cancer because the shared antigens are different between colon and melanoma, for example. So if you have a shared antigen fixed vaccine, it's better to restrict it to one disease where you know that most of the tumor cells will express this protein that is coded by the mRNA vaccine. So there are these different clinical studies, mostly by BioNTech and Moderna, recruiting either one specific type of cancer patients or broadly.
Okay, next question. Could mRNA vaccines be used to target microRNA induced by inflammation?
Could mRNA vaccines target? No, I would say no. So mRNA vaccines are made to code for proteins. So it does not have to, it does not, it cannot really affect the microRNA pathways. But speaking about inflammation, the mRNA vaccines can be used to deactivate the immune system. In multiple sclerosis, it's an autoimmune response. Your own immune system attacks the nerves. And Professor Ugo Sahin has shown in mice suffering from autoimmune disease similar to MS that you can vaccinate them, not to activate the lymphocytes, but to deactivate them. It's a different formulation, it's given intravenous. And then you specifically deactivate the pathogenic lymphocytes in autoimmune disease. So with the mRNA vaccine, you can vaccinate against virus, you can vaccinate against cancer, against allergy, but you can also devaccinate. You can, with special formulation of mRNA, modified in a liposome given intravenous, Professor Sahin has shown in the mice that you can de-immunize. And then this is a big hope that you can, with mRNA vaccines, treat autoimmune disease like lupus, like multiple sclerosis. So that's the reverse of vaccination, but it's still possible with mRNA in liposomes.
Ok, thank you. On a une question maintenant en français. Y a-t-il des conditions précises pour la prise du vaccin anticancéreux? Y a-t-il une population spécifique concernée ou est-ce que c'est disponible pour tout le monde?
Il y a ce qu'on appelle des critères d'exclusion et d'inclusion dans les essais cliniques. Donc, pour chaque essai clinique, il va y avoir une liste de critères d'inclusion. Il va falloir que le patient, par exemple, ait tel type de tumeur ou dans les essais cliniques de BioNTech et de Moderna qui recrutent solide cancer, presque tous les patients peuvent aller dans ces études, mais parfois, il y a quand même des facteurs d'exclusion qui peuvent être le grand âge, par exemple, parce qu'on sait que les personnes très âgées ont un système immunitaire affaibli et que chez ces personnes, la vaccination ne fonctionne pas ou peu. Donc, il y a parfois donc, des critères d'exclusion sur l'âge. Il y a parfois des critères d'exclusion sur les métastases. En particulier, les patients qui ont des métastases dans le cerveau sont parfois exclus d'essais cliniques parce qu'on sait que la réponse immunitaire ne va pas facilement dans le cerveau. Donc, il faut regarder sur chacun de ces essais cliniques de BioNTech ou Moderna. BioNTech et Moderna ont sur leur page web tous leurs essais cliniques. Vous pouvez aller voir. les paramètres de recrutement, et aussi vous pouvez leur écrire. Ils ont tous, les compagnies ont des emails sur leur page web pour les patients. Vous pouvez leur écrire, leur donner une description de votre maladie, et à ce moment-là, ils vont vous dire s'ils ont des essais cliniques qui seraient possibles pour vous ou pas, et selon aussi les endroits de recrutement. Mais effectivement, il y a toujours des critères de recrutement et des critères d'exclusion. Donc, il faut regarder selon son cas si vous pouvez rentrer dans certains essais cliniques de Moderna ou de BioNTech ou d'autres compagnies. Il faut à chaque fois regarder les critères ou écrire aux compagnies, leur donner votre profil et ils vont vous dire si vos critères correspondent pour certains de leurs essais cliniques.
Ok. Autre question. Est-ce qu'il y a des expériences utilisant des micro-ARN ou siRN pour cibler et inhiber les ARNM des oncogènes responsables du cancer?
Oui, donc depuis assez longtemps, les siRNA sont apparus en fin des années 90 et dès le début, ils ont été utilisés pour essayer de supprimer les oncogènes dans les cellules tumorales. Donc vous pouvez utiliser des siRNA qui vont détruire les ARN messagers codant pour des cellules, pour des protéines, pour des oncogènes, et à ce moment-là, réduire la prolifération des cellules et les induire en apoptose. Donc ça s'est développé depuis longtemps. Que je sache, ça n'a pas été encore approuvé. Alors, il y a aussi des antisens, des ARN antisens qui sont utilisés pour bloquer des ARN messagers codants pour des oncogènes. Donc, antisens et siRNA sont utilisés depuis assez longtemps contre les cancers. Que je sache, n'ont pas encore montré leur efficacité. Le problème dans ces cas-là, c'est que vous avez besoin de transfecter vos siRNA ou votre antisens dans toutes les cellules tumorales. S'il y a une cellule tumorale qui n'a pas son siRNA ou son mRNA, elle va continuer à proliférer. Donc, c'est un peu compliqué avec ces approches parce qu'il faut vraiment avoir des liposomes qui donnent le siRNA dans toutes les cellules tumorales et induisent toutes les cellules tumorales à mourir. Et c'est un peu le problème. Si vous avez un liposome qui donne à 90% des cellules tumorales le siRNA, 90% des cellules vont mourir, mais 10% vont continuer à croître et vous n'aurez pas d'efficacité clinique. Donc, c'est utilisé effectivement depuis longtemps, antisens et siRNA contre le cancer. Que je sache, ça n'a pas encore été validé, mais peut-être que des nouvelles formulations de liposomes vont permettre de valider les siRNA contre le cancer.
Okay, someone is asking us, for how long do we need to take this mRNA vaccines? Like, should we take it all our lives, or we can stop it once the cancer is cured?
Peut-être une dernière question pour ne pas beaucoup épuiser Dr Pascolo. Okay?
Non, c'est un plaisir de répondre aux questions.
Ouais.
Au contraire, pour moi, c'est aussi important de savoir quelles sont les questions que les gens se posent. Ça me permet aussi, dans mes prochaines présentations, d'anticiper et de présenter les aspects qui sont intéressants pour les gens. Donc, j'apprends, au travers des questions, j'apprends aussi. Donc,
thank you so much. Thank you.
Et c'est agréable. Donc, combien de temps? Pour l'instant, en général, dans les protocoles de vaccination par ARN messager contre le cancer, les patients sont vaccinés pendant tout le temps où la maladie ne progresse pas. Donc, pendant tout le temps, les patients sont stables au niveau du cancer, les tumeurs ne progressent pas à moins de 25% ou qu'elles régressent, vous pouvez vacciner. Normalement, quand les patients progressent, il faut arrêter le traitement expérimental. Ce n'est pas toujours le cas. Parfois, les patients progressent mais veulent continuer sur leur propre décision de continuer les injections. Alors, ça se fait hors protocole, ça s'appelle, c'est-à-dire que par le protocole, on arrête de les étudier. Par contre, par compassionate, par utilisation compassionnée, on continue éventuellement à injecter le patient si le patient désire continuer malgré l'évolution de son cancer, continuer à recevoir le vaccin, par exemple. Donc, normalement, par protocole, pendant que les patients répondent à la vaccination, qui sont stables ou qui régressent, on continue à vacciner tout le temps. Donc, alors, il peut y avoir peut-être quand même une limite de temps, mais en général, quand vous avez des patients qui répondent bien à votre traitement, vous continuez à injecter tout le temps.
Ok.
Par mois peut-être, ou une fois tous les 2 mois, mais vous continuez en général à les injecter.
Okay. Joel asked once again about the criteria to have these vaccines. I guess you answered it before. So Joel, if you didn't answer your question, please be more specific again in the comments. Fatima is asking, concerning cancer therapy, is using RNAi vaccines an option? If yes, is it more efficient than mRNA vaccines? mRNA vaccines targeting research mutant genes and a specific type of cancer. Excuse my ignorance in those terms.
But RNAi is not a vaccine. RNAi are reagents that will, as we spoke about before, so RNAi or siRNA, it can be also antisense. These are synthetic RNA oligonucleotides that will be made to block oncogene. And so this is not vaccine, this is therapy, it's like a medicament. Yeah, so you, with anti-cancer drug, you also block somehow the proliferation of tumor cells. And this is what the siRNA or the RNAi oligonucleotides, they do, they block proliferation. This is not vaccine. It's therapies, it's medicaments.
Next question. When mRNA are used as a vaccine against cancer, is it only to avoid relapse?
No, it's both. So you have clinical studies, as I have shown also. So you have patients who are tumor-free. Like if you have a melanoma, it's taken out, so you are tumor-free. But if your melanoma shows a high grade, we know that you have a high, that there's a high rate of recurrence. So you will suddenly have metastasis from the melanoma. And so it's the same for breast cancer. A lot of breast cancer are detected early. So the cancer is removed, so the patient is tumor-free. But the pathology may see that it's already infiltrated, so you have a high risk of having metastasis coming in the next 2 or 5 years. So there are these two categories of patients, patients who are tumor-free after operation. They have no more tumor, but high risk of relapse. So you can vaccinate these patients to prevent the relapse. That takes many years. You have to vaccinate them and to look after 5 years if they have relapsed or not. And you have patients who have metastasis. You can measure the metastasis in the liver, for example, by imaging, and then you vaccinate these patients and you look whether the metastasis regress. Because of course, if you have liver metastasis, you cannot operate, or most of the time you cannot operate. So you have a metastasis and then you treat the patient with mRNA vaccine to see the metastasis go down. So you have some kind of prophylactic, I would say, use of mRNA in tumor-free patients after operation. They are tumor-free, they get vaccinated, you are looking for their so-called tumor-free survival, so or relapse-free survival, so the time that they continue to live without having a relapse. And you have also patients with metastasis, measurable, you vaccinate them and you try to see if the metastasis regress, if no new metastasis come. So this is then stable disease. If they regress, it's partial response. If they totally disappear, it's complete response in these patients. So you have really the two possibilities of kind of prophylactic vaccinating people who have tumor-free, but high risk of relapse, and vaccinating people who have metastasis, and you vaccinate them in order to get rid, but the immune system destroys the metastasis.
Okay, thank you. Questions keep coming, but because of the lack of time, we will have to reduce it a bit. We will answer 2 more questions. And to be fair, I will give the questions of the people that still did not ask any questions. So first question, would it make sense to reconstitute the immune system before starting immune therapies, as most cancer patients have an impaired immune system?
Yes, and this is regenerative medicine in a way. So you would like, especially in elderly patients, as I said, they have a very weak immune system sometimes, and cancer patients have a weak immune system when they have metastasis. And ideally, you would rejuvenate their immune system before vaccinating them. And we work on that. We work on trying to rejuvenate the immune system. You know that over time, the thymus, which is the site where the T cells are produced, regress. So you have a big thymus when you are a kid, and then it's adult, it gets down. So, and the immune system when you are older is like memory cells. You have barely any naive cells that can be activated by vaccine against new antigens. So there is indeed research, and we perform this kind of research to regenerate the immune system in elderly, so that we would first make the immune system a little bit fresher before vaccinating. But you're right, it's a problem in elderly. That's why sometimes the clinical studies, they have this kind of age limit in vaccines, because we know that elderly patients have a weak immune response, and tumor patients with metastasis also have a weak immune response. So to vaccinate people who have a weak immune response, of course, is a problem. So you're right. I mean, to rejuvenate the immune system is a question. And with synthetic mRNA, you can do everything. You can also rejuvenate the immune system. We are trying to do that with mRNA. So it's not vaccine mRNA, it's mRNA for therapies. But ideally, indeed, you will rejuvenate the immune system before vaccinating elderly or advanced cancer patients. This is in development, in research.
Okay, thank you. We will take one last question. As you mentioned that unmodified mRNA works better than modified one against cancer. I wonder if it is because of innate immune activation or because of other reasons as well.
No, I didn't say it works better. I said Moderna used modified mRNA for its anti-cancer vaccine and has good results, as I've shown, good clinical responses. BioNTech for its anti-cancer vaccine used unmodified mRNA. The Moderna modified mRNA is given intramuscular, in liposome intramuscular. The BioNTech non-modified mRNA vaccine against cancer are given intravenous. So it really depends how you formulate your mRNA and how you inject it and the dose. So there are so many parameters, the antigen coded by the mRNA, whether the mRNA is modified, non-modified, in what kind of particles it is, and where it is injected. All these are parameters that have to be optimized. And if you have an optimized modified mRNA vaccine, it's good. If you have an optimized non-modified mRNA vaccine, it's good. So for the time being, BioNTech is non-modified against cancer, Moderna is modified against cancer, but both of them have really good immunological results and clinical results. So not that one is better than the other, they are both inducing immune responses against cancer and clinical responses. And it's really depend when you speak about modified versus non-modified, it's really depending what you do, if it is prophylactic, therapeutic vaccines, and how it is formulated. The advantage of the non-modified, the BioNTech formulation given intravenous, is that very quickly after injection, within 2 or 3 hours, the people make interferon, type 1 interferon, because the non-modified mRNA will trigger inflammation, systemic inflammation, and will trigger type 1 interferon, and type 1 interferon is good against cancer. So by the non-modified mRNA in liposome given intravenous by BioNTech, the patients immediately make type 1 interferon, which is already a first response against their cancer. And then they make an adaptive immune response, BNT cells, which again will attack cancer. So you have like a double effect of the liposomal formulation of non-modified mRNA by BioNTech. The Moderna modified mRNA against cancer given intramuscular, you maybe don't have the type 1 interferon induction, but you have other type of immunity induced, stronger T and B cells, which maybe then is interest. Maybe both should be combined, I don't know. So it's a lot of parameters that have to be optimized in order to know what mRNA is best, which sequence, what antigen, what modifications, what formulation, what site of injection, what dose, what schedule. All this has to be optimized. That's why we have such better results in the last 20 years. It's always getting better because we solve some of these issues so that the mRNA vaccines against cancer always get better.
Thank you so much, Professor Pasculo. C'est passionnant. What I suggest, Céline, if we can copy all the questions that we have in the chat box, we will be sending them to Professor Pasculo when he has time. Maybe he can help us to answer them. Don't hesitate to send us an email to tell us about topics, subject that you're interested in, so we can plan our future webinars. And with Professor Pasculo, we will continue our series of webinars as he promised us. So I thank you so much. I don't know if you want to conclude with something, Professor Pasculo.
I want to thank you for your organization, because it makes then really the possibility to communicate, to explain, and then people can decide for COVID vaccine, for cancer vaccines. I think to be didactic, to explain so that people understand. I think it's also fascinating, the mRNA world, so what mRNA does in cell, what synthetic mRNA can do. I think it's of interest, so thank you for making it available for people to participate, to understand, to listen, to try to understand, and to be capable and to think about all these technologies, maybe to participate for some of you who want to do research or medical research. And or to be a patient if you think you need such interventions. So thank you for organizing this kind of event. Thanks again to the Diane Foundation for making it possible and for the great work. And of course, I'm available to continue to explain. It's a nice opportunity for me. Also, I learned from your questions. So for me, it's also good for me. So thank you.
Thank you so much. Thank you so much.
Merci, Dr Pascolo.
Merci à vous, Diane.
Voilà, Diana, tu as envie de conclure, de dire quelque chose à la fin?
Tu parles avec qui?
Avec toi.
Moi, j'aimerais entendre le webinaire en français. J'ai beaucoup de mal à le comprendre en anglais.
On va peut-être alterner, peut-être faire certains webinaires en français, certains en anglais, c'est vrai.
Surtout que celui-là m'intéressait particulièrement, mais je n'ai pas pu tout suivre. C'était un peu difficile pour moi.
Oui.
Je peux le refaire en français.
Oui,
génial.
Bien. On pourrait peut-être pour un public francophone spécifier un webinaire en français. Ça, c'est une
ça serait super.
Ouais.
Merci beaucoup en tout cas.
Merci, allez, bonne soirée. Au revoir tout le monde et à bientôt.
Au revoir.
Au revoir depuis la Suisse. Bonne chance à vous.
Merci beaucoup. À bientôt, Steve. Ciao.
Au revoir.
Bye.